Hello, my name is Katarzyna Łepeta. I'm a postdoc in the lab of Markus Affolter at Biozentrum in Basel, and I would like to present you the synopsis of the approach for targeted protein phosphorylation that we developed in the lab. This work was recently published in the Journal of Cell Biology. Communication is everything. This is as true for us humans to get the correct message across as it is for cells. In order to form a complex organism and stay healthy, cells need to receive signals from the environment, communicate with other cells, and integrate the response within themselves. A group of enzymes, so-called protein kinases, plays a key role in internal cell communication. By attaching a phosphate group to a substrate protein, kinases orchestrate activation of complex signaling networks to control vital cell functions and maintain a state of balance needed for cell to survive and function correctly. Defective kinase networks have serious consequences, such as developmental disorders, cancer, or immune deficiency. Most kinases have multiple recipients. The vast complexity of the signaling networks poses severe challenges, and despite technical advances, it remains very challenging to understand single lanes of such intricate meshwork. Therefore, how to target a defined substrate of a given kinase? In other words, how to dissect a single chat from a group chat in cells? To address this need, we developed a method that provides means to direct the phosphorylation to a chosen tagged target in a living organism. For this goal, we coupled an activated catalytic domain of a chosen kinase to an antibody fragment, a nanobody. Such kinase, when co-expressed with a GFP-tagged substrate protein, will bind to it via the nanobody, activating the selected recipient protein and not the other targets, allowing to channel the kinase activity into a chosen pathway to better understand this single lane of cell communication. Our main protagonist is a fly embryo, a great model to study animal development. Thanks to fly genetics, we can compare side by side regions of the engineered and the control tissue. We used advanced microscopy to assess the phosphorylation levels and to follow tissue behavior in developing fly embryo. We successfully evaluated our tool on the example of engineered Rok kinase, showing that expression of synthetic Rok results in a robust phosphorylation of GFP-tagged myosin 2 regulatory light chain protein, called squash in flies, as indicated with the yellow arrows. Since squash protein is essential for the activation of myosin 2 complex, crucial for regulation of cell contractility, we next verified how this robust phosphorylation with engineered kinase influences mechanical properties of cells in developing fly embryo, in a morphogenetic process called dorsal closure. Here you can see the videos showing a control embryo on the left side and an embryo expressing engineered Rok kinase in stripes on the right, and I hope you can notice the altered distribution of squash GFP in the embryo expressing engineered kinase, leading to the formation of large clusters, disruption of the actomyosin cable, and a defective closure. Therefore, our tool allowed also to address the importance of balance between contraction and stretching for proper development of the embryo. Despite the encouraging results with our engineered Rok kinase, we were aware that expressing activated kinase at high levels can be eventually detrimental to the cell, and that selectivity of the engineered kinase toward the GFP fusion target is unlikely to be absolute. Therefore, to minimize any possible toxicity, we tried to restrict the kinase activity to the time and place where a GFP target is present, and used for this a destabilized variant of GFP nanobody. In the absence of a GFP-tagged substrate, the nanobody is destabilized, and the whole nanobody kinase fusion protein is degraded. Only if a GFP target is present, the synthetic kinase binds to it via the nanobody, allowing for efficient phosphorylation. In a similar set of experiments as shown previously, we were able to prove that the use of destabilized GFP nanobody created an optimized synthetic kinase variant, which is similarly efficient, yet importantly, innoxious in the absence of a GFP target. In summary, in the paper we show detailed characterization of engineered kinases based on two different enzymes, namely Rok and Src kinase with various protein binders, and show efficient phosphorylation of their respective GFP fusion targets, providing means to direct the phosphorylation to a chosen tagged protein in a living organism in a tissue-specific manner. This can help us and other scientists understand better the single communication lanes essential for regulating animal development, as well as interpret better the faulty communication in complex diseases. We presume that after careful optimization, This novel approach can be adapted to other kinases and targets in various genetic model systems. If you want to find out more about the paper, please check the link below and feel free to reach out to us if you have any questions. Thanks for tuning in. Bye!